Hello, my name is Justin Singer. I work in Dr. Jana Patton-Vogt's lab at Duquesne University, and today I'm going to talk about my project, which is called the role of Acyltransferase Gpc1 and antifungal susceptibility in Candida albicans. For a little bit of background, Candida albicans is a yeast species that's a fungal pathogen, and it kills about 400,000 people per year. And most of the drugs for this are focused on membrane synthesis, or causing some disturbance in the cell membrane. This is important because Gpc1 is a protein that was discovered by a graduate student in our lab, and it works in the PC deacylation-reacylation pathway, also known as PC drip, which you can see in figure 1. This involves taking a PC molecule, deacylating it, and then forming just the head of the lipid, and then it gets reacylated one more time by Gpc1 to LPC, and then it can get acylated one more time to go back into a PC molecule where it can be reincorporated back into the membrane. My project focused on knocking this gene out so we don't have this protein functioning and see if we observe any sensitivities to certain antifungal drugs. As figure 3 notes, most of these antifungals are targeting membrane synthesis or some type of phospholipid synthesis. And if we skip over to figure 4, you can see the first drug I tested, which is ketoconazole, which is inhibiting ergosterol synthesis, which is like a cholesterol but for yeast. And we noticed that we have an increased sensitivity in the Gpc1 knockout. So we have the wild type, which is the circle, and then we have the triangle, which is Gpc1 knockout, and the gray square is Gpc1 knockout plus Gpc1. So we knock the gene out and then reinsert it to make sure to serve as a double control. And we noticed an increased sensitivity in the knockout when we treat it with the drug. So we noticed some sensitivity there. And we did a similar assay in figure 5 with myriosin, which is inhibiting sphingolipid synthesis. In this assay, we kind of did a very high concentration for only an hour, and then we got rid of the drug and then let them grow up to see if they could almost recover from this drug treatment. And as you can see again in the Gpc1 knockout, we see a greatly increased sensitivity. And as far as figure 6, myriosin, or miltefosine, doesn't exactly have a known use. So we noticed that the structure looked very similar to lyso-PC, which is LPC, otherwise known as the product of Gpc1 and Gpc. So we decided to use that as a drug, and sure enough, it was an extremely stark difference when compared with the wild type and the Gpc1 knockout plus reintegrant. And from this data, we can make the conclusions that Gpc1 knockouts are more sensitive to the inhibition of ergosterol synthesis by ketoconazole. Gpc1 is more sensitive to, or knockouts are more sensitive to sphingolipid synthesis, and also general glycerophospholipid synthesis via miltefosine inhibition. Some things I'd like to look at in the future is look at other classes that I didn't get to examine, like echinocandins and other different azole classes, because those seem to have more promising data as a whole, and look at more miltefosine stuff, such as expression change or maybe an ROS study, something like that. I would like to acknowledge Dr. Bernard Hubé for donating Candida strains, and the National Institute of Health grant, and Dr. Jana Patton-Vogt for funding. Thank you.